In last class, we had a quiz, right? I mean, the Sunday
Yes, sir.
class was, yeah, ask a keyboard.
So
Tuesday.
Tuesday.
Tuesday, okay. So last Sunday class I cancelled it, and the class before that we had a quiz. The quiz question was easy, I guess. It was not difficult. So okay, we'll discuss about the quiz questions next class. So before that, the next class is next Sunday. Yeah, okay. So before that Sunday, I can provide you both the quiz and the meet script. So we can discuss about those in that class. Okay, so in last class, I discussed about this lysogenic and lytic cycles. So you have questions from here?
No, sir. আপাতত no questions.
আপাতত no questions. Okay. আচ্ছা, অ্যাসাইনমেন্টটা কি তোমরা বুঝতে পারছো?
স্যার এটা কি আমাদের নিজের রিসার্চ করে করতে হবে?
হ্যাঁ, একটু সার্চ করো আর কি। হ্যাঁ, আচ্ছা আমি দাঁড়াও, তারপর একটু অ্যাসাইনমেন্টের একটু ওপেন করি। আমি তোমাদের হালকা আইডিয়া দিই। আচ্ছা, অ্যাসাইনমেন্টের যেটা ছিল তোমাদের, I mean, for the assignment there was 3 questions, right? So the first question was like protozoal disease.
জি স্যার।
Okay, for protozoa, we have a lecture on protozoa. So during that time I can explain the protozoa. I mean, usually, I mean, whenever we think about microorganisms, basically most of the time we think about bacteria and virus more frequently. These two microorganisms is discussed more frequently, bacteria and virus. But beside the bacteria and virus, protozoa is also protozoa are also causative agent for several deadly diseases. Okay. So if you Google the protozoa is a microorganism, you already know, and protozoa cause several diseases, deadly diseases. Okay, one is like malaria. Malaria is caused by Plasmodium vivax, right? It's Plasmodium vivax is a protozoa or bacteria. Let me confirm. So the causative agent of malaria, Plasmodium vivax, it's a protozoa. It's not a bacteria. It's not a virus. It's a protozoa. Okay, and you know malaria is a deadly disease. So there are several other protozoal diseases. So in this, I mean the assignment, the question number 1, please find 5 protozoal diseases and their causative agent. Causative agent means like the malaria. It's a protozoal disease caused by Plasmodium vivax, and then write about how they transmit. Transmission is an important thing for infectious disease spread. মানে কি হবে ডিজিজটা ছড়ায় আর কি। যেমন you know how malaria transmit through mosquito, right?
Female Anopheles mosquito.
Yeah, and then how to prevent and treat protozoal disease. So for example, what do you mean by prevent? Say for example, if we want to prevent malaria, so basically when we say that how to prevent vaccine, I mean we think about vaccination. So if we can get a vaccine, then we can prevent the disease. But for like malaria, there is no prevention. I mean, no vaccination. Till now, there is no vaccine for malaria. Then how we can prevent malaria? Till now, the most, you can say the successful or practiced method is to prevent the mosquito. As there is no vaccine for plasmodium virus, I mean vaccine for malaria. So the best way to prevent malaria is to prevent the mosquito. Okay, so if say you find a protozoal disease and for that vaccine is available, then you can write, okay, so we can prevent it by vaccination. Okay, and treatment is medicine. Prevention and treatment is different thing, right?
Yes, sir.
Treatment is
Yes, sir.
medicine. Okay, so this is the things you have to write in this first assignment. And the second one is algae. We have a chapter on algae actually, also another lecture of algae. So I'll also briefly discuss about that lecture. And in the second one, you just have to, I mean, algae. Okay, there are many algae. They are beneficial. Okay, they do lots of beneficial things for us. Please try to find those beneficial things. It will not be that difficult. If you Google it, you can easily find the information. And this is a good practice. I mean, in this current world, there are lots of information out there. So it is important to learn how to find out the information. Okay. So usually for scientific things, we always check published article. But I can understand you are fresher. So for you, I mean, finding information by reading the published scientific article will be difficult. I can understand that. So for you, it's not necessary that you will check the published article. You can find the information. I mean, if you find the information from just, for example, a science magazine or just newspaper or just in the Google, it will be fine. Okay. So try to find some beneficial role of algae. Okay. And in the third one, this one is about bacteriophage. Bacteriophage is a virus or bacteria? So
It's a virus.
virus, yeah, exactly. It's a virus. And what is its speciality?
So they can eat up virus. Sorry, bacterias.
Yeah,
They
okay,
can
we can say
they
that.
can
Yeah,
destroy bacteria.
basically they like bacteriophage are the virus for bacteria. So they infect bacteria and kill it. Okay. So based on this capacity of or capability of bacteriophage, we can use them as treatment. Well, so far it is not a regular treatment, and so far there is like maybe only few cases, like 2 or 3 cases where bacteriophage was successfully used to treat disease. So what kind of disease and why? I mean, use of bacteriophage can be is a promising is I mean it actually I mean bacteriophage it can be a good treatment in the near future to treat infectious disease. What kind of infectious disease and why we need to use bacteriophage? So you all
Sir,
know about the drug resistance thing, right? So you have heard this word like the
antibiotic resistance. জি স্যার।
antimicrobial resistance. শুনছো তো না?
জি স্যার। So যখন bacteria become resistant এই antibiotics, they lose their resistance to bacteriophage. আর যখন তার resistance টা grow করে for the bacteriophage, তখন they lose their resistance to antibiotics. So
Okay. You're okay. So you're like partially right. Okay. So let me explain. So antimicrobial resistance means the bacteria they are getting resistance against the available antibiotics. This is called antimicrobial resistance. That means the okay. When we say antimicrobial resistance, it refers to all the microorganisms, not only bacteria. Actually, also other microorganisms like protozoa, uh, the fungi, fungus also can cause disease, bacteria, even virus. they are getting resistance against the available medicine or drug. Okay. So, and for bacteria, we usually say antibacterial resistance. Okay. So, antibiotic resistance. For when we say about bacteria, we usually say antibiotic resistance. So, almost all the pathogenic bacteria, they are developing resistance against the antibiotics. So, this is a serious issue, and it can be a devastating thing for us. So, like say, if I mean nowadays, if we get infected by bacteria, we take antibiotic, we get cured. Okay, but if the bacteria get resistance, then what will be the problem? We may die just of a simple infection, simple fever. We can die of, right? Because no available antibiotic will work against it. It's actually a very serious issue nowadays because lots of pathogenic bacteria they are getting multi-drug resistance, and yet and there are some bacteria already there out there who are resistant against all the antibiotics. So now how then, if the bacteria resistance against all the antibiotics, how we can treat them, how we can kill those bacteria, those pathogenic bacteria if we get infected? So here comes the bacteriophage. Bacteriophage are bacteria specific. Like the same bacteriophage will not infect all the bacteria. Say for example, E. coli. The bacteriophage for E. coli is specific. They only infect E. coli. So Vibrio cholerae is a bacteria. The bacteriophage who infect by Vibrio cholerae, they will infect only Vibrio cholerae. They are very specific. Actually, they are more specific. What I mean by more specific, we know E. coli is a bacteria and it has several strains. There are many bacteriophage who are strain specific. So, for example, there are some bacteriophage who don't infect all the strain of E. coli. They only infect a particular strain. So, we can use bacteriophage to kill bacteria, to kill multi-drug resistance bacteria. Okay. So, in this number 3 assignment, you have to write about this thing, and this is it has a name. I mean, use of bacteriophage to kill uh, drug resistance bacteria. It has a name. It is called phage therapy. Did I write this here? A phage therapy kothao likhi? Okay, I didn't write about this in the question, but this is actually called phage therapy. So in this assignment number 3, you have to write about the phage therapy. Okay.
Okay, sir.
So try to find out this information by Googling them. আচ্ছা অ্যাসাইনমেন্ট নিয়ে কারো কোনো কোশ্চেন আছে? এনি কনফিউশন অ্যাবাউট দ্য অ্যাসাইনমেন্ট?
Sir, you have said that, sir, we can learn about protozoas and algae in other lectures.
Yeah,
Are there in this course?
yeah, in this course.
Okay, sir.
Mike 1-1. Okay. Okay, so if anyone don't have any questions from the assignment, then we can move back to our study. I have already discussed about the life cycle of virus, two life cycle, lytic and lysogenic cycle in the previous lecture. And also I've discussed, when the bacteria will enter, when the virus enter the lysogenic cycle and when the lytic cycle. Today, I'll mainly discuss about the one-step growth curve. So like when a virus infect its host, its growth follow a pattern. It's almost like the bacterial growth curve. You guys can remember the bacterial growth curve, right?
Yes, sir.
In the bacterial growth, there are several steps. Log phase, no, no, sorry, lag phase, log phase, stationary phase, dead phase. Okay, so virus also follow a pattern during their growth, and if we plot that pattern in a graph, or if we try to graphically show that their growth, they look like this. Okay, like this graph, and it is called one step growth curve. Okay, so let's try to understand what one step growth curve is. Okay, now think of a scenario. Say a virus, it has a certain amount, a virus, it has infect its host. So usually in the beginning, a particular amount of virus infects the host. So for example, you want to graphically present this. So you put you take a graph, and in x-axis, you plot the time, and in y-axis, you plot the number of virus. Okay, so say in the beginning, one log number of virus. So this amount of virus infect the cell. Then what will happen? After the virus infect the cell, their number will increase, right? So if this is the beginning point, then the virus should, this amount of virus infected the host. Say here the host is bacteria. So this amount of virus has infected the bacteria. Then after infecting the bacteria, they will multiply inside the bacteria, right? So their number should increase. So why it has not increased here? We can see that the number has decreased and number has become 0. And a certain time period, the number of virus remain 0. Can you guys say why it is like this? মানে এরকম কেন হলো? Anyone?
স্যার কোয়েশ্চেনটা রিপিট করা যাবে?
আচ্ছা, যেটা হচ্ছে ধরো এই অ্যামাউন্ট অফ ভাইরাসটা, তোমরা তো গ্রাফটা দেখতে পাচ্ছ না?
জি স্যার।
X axis is time, y axis number of virus. Say this amount of virus has infected the host cell. So after infecting the host cell, the virus, they replicate or increase their number inside the host cell. So in the graph, the number of virus should increase, right?
Yes.
But instead of that, we can see that the number of virus has decreased. And the number of virus is here is 0. এখানে ভাইরাসের সংখ্যা কি শূন্য না?
জি স্যার।
And they remain 0 for a several time period. A certain time period, the number of virus remain 0. Why? এরকম শূন্য আছে কেন? এটা তো বাড়ার কথা। Any wild guess?
স্যার বায়োসিনথেসিসে শুধু ভাইরাসের নিউক্লিয়াস ভিতরে গেছিল, এখানে 0 থেকে 1 মানে 1 থেকে 0 হয়ে গেছে এর জন্য।
Okay.
স্যার ভাইরাস টেক ওভার করার সময় তো they just inject their DNA material into the host cell, right? The আসল ভাইরাসটা তো মারা যাচ্ছে once you inject the DNA material in. So that's why the number of viruses decreases, because that one virus or the DNA material already host ভিতরে ঢুকে দিয়েছে. So now that it's dead, ওই DNA টা যতক্ষণ লাগবে to take over, that's the biosynthesis and maturation part, I think.
Okay, you guys are almost correct. Yeah, both of you are almost correct. Yeah, that's happened. The virus, this amount of virus has infected the host cell, and the whole virus didn't enter into the host cell. They injected only their nucleic acid. So when the nucleic acid of virus is inside the cell, the different parts of the virus get synthesized, like the viral nucleic acid. The number of viral nucleic acid increased through synthesis. Also, the proteins, that is the building block of virus, they get synthesized. But during that time, it's not a complete virus, right? DNA, protein, they're separated.
Yes sir.
They are not a complete infectious virus. So that's why theoretically, although inside the cell there are many parts of the virus, but there is no complete virus which can infect other cell. So that's why the number remains 0. মানে ভাইরাস যখন ইনফেক্ট করে একটা হোস্টকে, সে তো শুধু তার নিউক্লিক অ্যাসিডটা ইনজেক্ট করে দেয়। তো নিউক্লিক অ্যাসিড সেলের ভেতরে যাওয়ার পরে কি হয়, সেলের ভেতরে হচ্ছে নিউক্লিক অ্যাসিডটার সংখ্যা বাড়তে থাকে, তারপরে হচ্ছে রেপ্লিকেশনের মাধ্যমে, তারপর হচ্ছে ওই যে ভাইরাসের যে বডির যে আদার পার্ট গুলা থাকে, যে প্রোটিন ফর এক্সাম্পল, সেগুলো হচ্ছে সিনথেসিস হতে থাকে। কিন্তু সেখানে সেলের ভিতরে কিন্তু কোনো কমপ্লিট ভাইরাস থাকে না, তার শুধু বডির আলাদা আলাদা পার্ট গুলা থাকে। সো থিওরিটিক্যালি কি, নাম্বার তো 0, দেয়ার ইজ নো কমপ্লিট ভাইরাস ইনসাইড দ্য সেল, সো নাম্বার হচ্ছে 0 থাকে। ওকে, সো okay, so this time period, while when the different body parts of the virus, nucleic acid, the protein, get synthesized, which we call biosynthesis, during that time there is no complete virus, so that's why the number of total virus will remain 0. Okay, so
Okay, sir.
this has a name, this time period, it is called eclipse period. So first the virus infect the host, okay, and they inject the nucleic acid in the host cell. Okay, so this is called penetration, and then they synthesize. Just one second. Okay, I got a call. Just one second. Hello. ওয়ালাইকুম আসসালাম। আমি যে একটু ক্লাসে আছি। ওকে, হ্যাঁ ঠিক আছে। Okay, guys, sorry, I'm back. Okay, then this time period is called penetration, and then when the viral particles get synthesized, this period is called eclipse period. So once all the viral particle is synthesized, then the different particles, they assembled and become a full virus, and this is called maturation. After the biosynthesis of different viral part, they get assembled and become a full viral particle, which is called that means the maturation occurs, and then all those matured virus, they get released from the cell, the host cell. Basically they disrupt the host cell and get released. So that means, and this is called bursting. Okay, so burst, ভাইরাস হোস্ট সেলটা বার্স্ট করে ভাইরাস গুলো রিলিজ হয়ে যায় আর কি। সো in this time a big amount of virus, okay, a certain amount of virus, they get released from the cell. So suddenly the number of virus will increase. Okay, so that's why we can see the increase of number of the viruses. Okay, so this is called burst size. The number of virus which has been released upon lysis of the host cell is called burst size, and the time required for this is called burst time. Okay, so together this graph is called one-step growth curve. Did you understand now why the curve look like this? বুঝছো তো নাকি কার্ভটা কেন এরকম দেখায়?
জি স্যার।
না বুঝলে অসুবিধা নাই, বলতে পারো, I can explain again, no problem.
Sir, is there any specific reason that the eclipse period was named?
Okay, I think eclipse এর মানে কি, eclipse এর এক্সাক্ট ইংলিশ মানে কি?
স্যার বাংলাটা বলতে পারি, উপবৃত্ত।
না, eclipse এর আলাদা আলাদা মানে আছে। দেখি আমরা eclipse এর আরো কিছু মানে দেখার চেষ্টা করি। what is the meaning of eclipse, this English word? Okay, first during is the distinctive marking.
Sir, eclipse মানে ভাইরাসের eclipse period নাকি just এমনি wordটা eclipse?
Basically, I mean, why it has been named eclipse. Okay, so this period is called eclipse period, but why it has been named eclipse? ওকে, এক্লিপস মানে একজন বললো যে আমরা জানি হচ্ছে কি এক্লিপস মানে কি জানি, এটা কি বলে, চন্দ্রগ্রহণ না সূর্যগ্রহণ কি জানি বলে না?
জি স্যার।
ওকে, বাট এক্লিপস হ্যাজ অ্যানাদার মিনিং অ্যাকচুয়ালি।
স্যার এক্লিপস হচ্ছে ওইটা না যখন দ্য মুন কভার্স দ্য সান,
Okay,
মানে একটা পিরিয়ডে
it is actually one meaning of eclipse. This eclipse word has another meaning. Okay.
স্যার আরেকটা হচ্ছে যে মানে অনেক র্যাপিডলি, মানে কি বলবো, অবসকিউর হওয়া, মানে একজন অনেক পপুলার ছিল তারপর তার রেপুটেশন অনেক তাড়াতাড়ি খারাপ হয়ে যায়, এটা
আচ্ছা,
হচ্ছে আরেকটা এক্লিপস।
হ্যাঁ, eclipse এর আরো মিনিং আছে কিন্তু। So if you Google the word eclipse, you can see that there are several meanings. That one, it has one meaning: a phase during which the distinctive markings of a bird, especially a male duck, are obscured by molting of the breeding plumage. মানে ওই একটা হাঁসের, তোমরা গুগল করলে দেখবা যে কিছু মেইল ডাক আছে, তাদের মার্কিং গুলা ওই সময় হচ্ছে ইয়া থাকে আর কি, তোমার অবস্কিউট থাকে, মার্কিং গুলো দেখা যায় না আর কি, হাঁস থাকে, হাঁসের মার্কিং নাই মেইল ডাকে, হ্যাঁ। অথবা তোমার এটা আরেকটা মানে আছে যে অবস্কিউর অর ব্লক আউট। অবস্কিউর, অবস্কিউর মানে কি, যে ওই সময় আসলে কোনো কিছু ক্লিয়ার না, ঠিক আছে, কাইন্ড অফ ওখান থেকেই না এই পিরিয়ডটার নাম এক্লিপস পিরিয়ড দিতেছে আর কি। I mean, during the eclipse period, eclipse, things happens, but we don't see it. Okay, I mean, another meaning of eclipse is obscure.
Yes sir.
Okay, so
Darkness is
yeah,
also
kind of, yeah, you can see that the darkness or block out. I
yes
mean,
sir.
what happened during eclipse? It's get blackout. So basically, I mean, they just named it because of that. It's just kind of name that in this eclipse period, it's kind of blackout. We don't see any virus at all. It's kind of blackout. Okay. সেখান থেকেই মেবি আই থিঙ্ক নামটা দেয়া হয়েছে আর কি, আদার কোনো কারণ নেই। ওকে,
জি স্যার, বুঝতে পারছি।
okay, so you understood the curve, right?
Yes sir.
Okay, so this curve has actually significance for pathogenesis cells, like say for yeah.
কার্ভটা আবার স্ট্রেট হয়ে যায় কেন
Oh,
একদম শেষে?
so straight হয়ে যায় কেন? Say for example, one virus, it has infected or say virus has infected the host cell. So there is limit that how many new virus can be formed inside a host cell, right? Or say for example, there was 10 host cell, okay? And all the 10 host cell get infected by a virus. Okay, then inside those 10 host cell, the number of virus got increased. So it has a limit, right? Say for example, if I mean, so okay, so first think about one cell. A virus has infected the host cell, and inside them they have increased their number. So for every particular virus and the particular host, there is a limit that how many virus can be synthesized inside a host cell. Okay, so depending on the virus, it can be, the number can be ranges from 50 to 200. Okay, so the increase of number should be certain, right? So for say now, okay, so you understand about the one cell. Now think about 10 cell. So there was 10 host cell and all got infected by the virus. Okay, and from 10 host cell, a certain amount of virus can be released, not more than that, right? So this is the, you can say the kind of the maximum number which will released from all the host cell. তোমার হোস্টের ভেতরে ধরো ভাইরাস যখন হোস্টকে ইনফেক্ট করে, একটা হোস্টের ভেতরে কি পরিমাণ ভাইরাস রেপ্লিকেট হতে পারবে, মানে নতুন ভাইরাস তৈরি হতে পারবে, এটা তো লিমিটেড, এটা তো ডিফাইন্ড আসলে। সো এই নাম্বারটাও কি হবে, এটা লিমিটেড নাম্বারই হবে না। ধরো এই ক্ষেত্রে বাস্ট সাইজ হচ্ছে কত, এর মাঝামাঝি আর কি, ভাইরাসের উপর ডিপেন্ড করে এটা কম হতে পারে বা বেশি হতে পারে, বাট এই নাম্বারটা তো কি একটা ফিক্সডই হবে, ইনফাইনাইট নাম্বার তো হতে পারে না কখনো বাস্ট সাইজ, রাইট? চিন্তা করে দেখো হতে পারে? This is the number of virus which get released from the host cell upon lysis of the host cell. So this number will be limited, not infinite. It cannot increase infinitely, right?
তো স্যার এই নাম্বারটা আবার বাড়বে না when they start infecting more? So আবার
Yeah,
একটু ডিক্রিজ করবে, তারপর আরো ইনক্রিজ করবে?
yeah, you are right. You're absolutely right. So if you actually try to continue the graph, again, it should increase, okay? But for this graph, I mean, we don't make it that long. It's for like, you can say just one cycle. So তুমি
জি স্যার।
যেটা বলছো সেটা হচ্ছে next cycle. Yeah, of course. In the next cycle, it will be like that. I mean, the same cycle will get repeated, right? So for example, this amount of virus, they infected new host cell. So again, their number will become, so for example, in this period, there are like say around, what will be this one? 500. Okay, so there is 500 virus in this in during this time, and this all those 500 virus infected the host cell. Then the number will again become 0. There will be eclipse period, then burst time. That means the virus will get released, and again a certain, definitely a certain amount of virus can released. Okay, বুঝতে পারছো? So you can say that this is one cycle. In the next cycle, the same things will be repeated. একই জিনিস রিপিট হতে থাকবে পরবর্তী সাইকেলে, ঠিক আছে?
জি স্যার, বুঝতে পেরেছি।
Okay, so okay, so this was the one-step growth curve. Now, where is this? Okay, so I think you already know that. So I mean, I have already told you that for the bacteriophage, I mean, the virus will infect the, okay, not only for bacteriophage, usually most of the viruses, they are host specific. Okay, so for example, COVID-19. COVID-19 is a coronavirus. COVID-19, I mean, it's a one a strain of coronavirus, which is named SARS-CoV-2. COVID-19 is specific for human. COVID-19 is not going to infect any other animal at all, species at all. Okay. Okay. So then, like say for example, even bacteriophage, which I told in today earlier, that bacteriophage, they are also host specific. The bacteriophage which infect E. coli, it will not infect in any other bacteria or animal or human or plant. No. So okay, then another example. Say for example, you all heard the name HIV, right? HIV cause AIDS. AIDS is the disease. AIDS is not a virus. HIV is the virus. You know this thing, right?
Yes,
Okay,
sir.
I'll
জি
come
স্যার।
back. So HIV is a virus which is human specific. HIV will not infect any other species. So there is another HIV like virus which is not HIV. It is called SIV. Hmm. SIV, not HIV. SIV. SIV is a HIV like virus, but it is specific for chimpanzee. Okay, it's in fact only the chimpanzee, not the human. So all the viruses, they are host specific usually. Okay, they are host specific. Okay. So okay, so we're done with that now. For various, I mean, we know how to culture bacteria. কিভাবে ব্যাকটেরিয়া কালচার করতে হয় আমরা জানি না?
জি স্যার।
Okay, so we can also culture virus. So how we can culture virus? So if we want to culture bacteria, what we usually do, we prepare nutrient media. We can prepare nutrient liquid media or agar media, that means solid media, and we culture the bacteria over them. So can we culture virus on the nutrient media? What do you guys think?
নো স্যার।
Yeah, exactly. Why? Why we cannot culture bacteria virus on the nutrient agar or nutrient media?
স্যার ভাইরাস নিড সেলস অর হোস্ট।
Exactly. Virus
তো
need cells or host.
ইয়েস।
So that's why if we want to culture virus, we cannot culture them on agar media, but we can culture them on host cell. So in general, what we have to do if we want to culture virus? First, we have to culture the host cell. We can culture cell, right? So first we can culture the host cell, then we'll add the virus into that host cell. So then the virus will grow on the host cell. So this is the basic way how we can culture virus. মানে আমরা যদি ভাইরাস কালচার করতে চাই ল্যাবরেটরিতে, ভাইরাসটা নিউট্রিয়েন্ট মিডিয়াতে গ্রো করা যাবে না। সো আমরা যেটা করি, আমরা প্রথমে হচ্ছে সেলটা আমাদেরকে কালচার করে নিতে হয়। প্রথমে আমরা সেল কালচার করে সেলটা গ্রো করাই, ওই প্লেটের মধ্যেই করাই। দ্য প্লেট লুক এক্স্যাক্টলি সেম। দ্য প্লেট উই ইউজ ফর কালচারিং ব্যাকটেরিয়া and the plate we use for culturing cell, they look same. Okay, there is a very subtle differences. Okay, you don't need to know about the differences right now. When you will learn about cell culture, then you can learn about that. Just right now, remember that this plate look exactly same. So on the plate, first we have, if we want to culture virus on the plate, first we will culture the cell, then we'll add the virus on that cell. Okay, sir? Then the virus will grow on those cells. This is how we can culture viruses. So now, say for example, you want to culture bacteriophage. So what we have to do? You know, right? কি করতে হবে? আমরা যদি ব্যাকটেরিয়াল ভাইরাস মানে ব্যাকটেরিওফাজ কালচার করতে চাই, তাহলে আমাদের কি করতে হবে?
স্যার ইকোলিকে আমাদেরকে
আচ্ছা, তুমি ইকোলাই বলতেছো? ওকে, ঠিক আছে, ধরো তোমার হচ্ছে ব্যাকটেরিওফাজ ইকোলাই স্পেসিফিক। ওকে, ইওর ব্যাকটেরিয়া ইজ ইকোলাই স্পেসিফিক। সো হোয়াট ইউ হ্যাভ টু ডু? হ্যাঁ, বলো, এখন বলো।
So we have to culture E. coli
Yeah,
in culture media. Then we have
to
to
add the that bacteriophage on the ইকোলাই। এটা তো বলছো নাকি?
and
যে প্রথমে আমরা কালচার মিডিয়াতে হচ্ছে ইকোলাইটাকে গ্রো করাবো, তারপর ইকোলাইয়ের মধ্যে এই ভাইরাসটা দিয়ে দিব।
yes sir.
Exactly. Exactly this in this way we can culture bacterial virus. That means, say for example, you have a bacteriophage which is E. coli specific, and you want to culture this bacteriophage on the E. coli. So what you have to do? First, prepare a culture media, and on that media, inoculate the E. coli. You can do inoculate by, so okay, in this case, yes, in this case you cannot do strict plate method. Okay, you have to grow the E. coli by, say, a spread plate method. A spread plate method will be better choice. Okay, not in strict plate method. Okay, so you will prepare the culture media and grow the E. coli on the culture media by spread plate method, and you have to inoculate enough number of E. coli on the plate so that in the next day it become matte. The E. coli grow all over the plate. So please remember these things. Okay, so you want to culture E. coli specific bacteriophage. So first, prepare a culture media and inoculate E. coli, enough number of E. coli on it, and incubate it. So as you are using E. coli, you can incubate it at 37 degrees Celsius for overnight, like say 18 hours or 24 hours. Okay, so what will happen in the next day? The E. coli will grow all over the plate. They will cover all the surface of the E. coli. Then this is called mat. Okay, যখন এরকম পুরো প্লেটের পুরো সারফেস জুড়ে হচ্ছে কোনো ব্যাকটেরিয়া গ্রো করে থাকে, তখন সেটাকে আমরা ম্যাট বলি। ওকে, আই মিন ইট উইল ফর্ম এ ম্যাট। দ্য ইকোলাই উইল বি অল ওভার দ্য all over the surface of the plate. Okay, so then add your virus, the bacteria, E. coli specific bacteriophage over it, and then again incubate it. So in the next day, if you observe the plate, you will see this kind of things. So this is the plate. Okay, this is actually a photo of a real plate. So these are the bacteria, and you can see this spot. This spot, the virus, they have grown in. They have actually they made this spot. So well, so I think in this figure it's kind of difficult to see, but if you see the real plate, you will see that, observe that these spots, they are clear. So like these places where there is no spot, these are not clear, these are opaque. Okay, they will be opaque, মানে এগুলা ক্লিয়ার থাকবে না, এগুলা থাকবে ঘোলা। তুমি দেখেই বুঝবা যে এখানে ব্যাকটেরিয়া গ্রো করে আছে। হ্যাঁ, লাইটের এগেনস্টে ধরলেই বোঝা যায়। But this spot, they will look clear. I mean, if you observe them, I mean, put them against the light, you can see that this spot, they are clear, transparent. Okay, so these are the place where the virus has grown. Okay, এই স্পট গুলোতেই ভাইরাস গ্রো করছে। Okay, so now why they form this kind of clear zone, transparent zone or a spot? Because the bacteriophage, I mean the E. coli specific bacteriophage, they infect the E. coli and they lyse it. Okay, so that's why the bacteria which were here, they are dead. So the area, it became transparent or clear. Okay, so this is the way how we can culture the viruses. Okay, so in this slide they have just I mean it is I mean these two slide, it shows how we can culture bacterial viruses or bacteriophage, but the basic is same for all the viruses. Say for example you want to culture a human virus, what you have to do? Okay,
আচ্ছা স্যার, ওটার আগে আমি একটা কোয়েশ্চেন দিয়েছি।
yeah, sure.
আমাদের সেফটি প্রিকশনস কি থাকে যদি বাই অ্যাক্সিডেন্ট আমরা ভাইরাসটা কালচার করার সময় মনে করেন ওই স্যাম্পলটা হাত থেকে পড়ে যায়, তারপর শ্যাটার করে আর তারপর ভাইরাসটা ছড়িয়ে ছড়িয়ে যায়, সো তখন
Okay, good question. তুমি একটা ভালো কোশ্চেন জিজ্ঞেস করছো। So it's really a good question, that what will be the safety precautions if we want to work with virus? It depends on the virus. Say for example, okay, say for example you are working with bacteriophage. Bacteriophage don't infect human, right? They infect the bacteria, so we are kind of safe. We don't have to take that much safety precautions if we want to deal with bacteriophage. So you are working with the bacteriophage and you spill the bacteriophage. Okay, you don't have to worry. Just put some disinfectant there. You can put some hypochlorites. Okay, so some bleaching. The virus will be dead. We don't have to maintain that much safety precautions when we work with bacteriophage, because they don't infect human. Okay, now say for example you want to work with COVID-19 and you want to culture COVID-19, you have to use maximum amount of precaution for that. Why? Because COVID-19 is a dangerous virus, it is highly infectious, it easily infect human, and they spread through droplet, through the air. বাতাসের মাধ্যমে, ড্রপলেটের মাধ্যমে কোভিড-19 স্প্রেড করতে পারে। তো এই জন্য যখন আমরা কোভিড-19 কালচার করতে চাই, আমাদের হচ্ছে ম্যাক্সিমাম প্রিকশন মেনটেন করতে হবে। Okay, so now another example, say for example HIV. HIV is also a very dangerous virus, right? If anyone get infected by HIV, they may develop AIDS in near future. Okay, usually it may takes around 10 years. Usually in average it takes around 10 years. Anyway, but HIV is a dangerous virus. So if we want to, again, if you want to culture with HIV, we have to take a very high amount of safety precautions. But if you compare against COVID-19, I mean, if you compare between COVID-19 and HIV, the safety precautions required for COVID-19 is high than HIV. আর আমরা যদি কালচার করতে চাই COVID-19 এবং HIV, COVID-19 এর জন্য সেফটি প্রিকশন বেশি নিতে হবে HIV এর তুলনায়। Can anyone guess why? I mean, I will give the answer, but can anyone you guess why?
So HIV is only transmissible through body fluids, whereas COVID can be transmitted through nasal cavity or droplets. তো আরো মানে infectious বলতে more easily transmissible.
Exactly. Yeah. I mean, COVID-19 it's very easily it can easily transmit through the air, right? Through droplet. মানে বাতাসের মাধ্যমে COVID-19 কিন্তু খুব সহজে স্প্রেড করতে পারে ওই যে ড্রপলেটের মাধ্যমে, রাইট? কিন্তু এইচআইভি কিন্তু এইভাবে স্প্রেড করতে পারে না। HIV cannot spread through the droplets or air. HIV can spread only through body fluids. Okay. HIV is not a contagious disease. Okay, contagious. Okay, it's a contagious. Okay, আমরা সোয়াচে রোগ বলি না, সোয়াচে বাংলায়। HIV তো সোয়াচে রোগ না। HIV is not a contagious disease, right? But COVID-19 is. Okay, so that's why although HIV, the disease caused by HIV is more dangerous than AIDS is more dangerous than COVID-19 infection, but when we culture the viruses, the HIV, the safety precaution is less than COVID-19. But against the safety precautions have to be quite high if we compare with the bacteriophage virus. Well, indeed there are, depending on the safety precautions, there are 4 types of lab in this world. And we name them as BSL1, biosafety level 1, 2, 3, 4. Okay, there are 4 level of lab. 1, 2, 3, 4. For COVID-19, we need safety level 4 lab. For HIV culture, we need safety level 3 lab. Okay. For safety level 2, okay, I forget the example. For say, for example, bacteriophage, we don't need any safety level at all. We can use safety level 1, BSL1. Depending on the virus, we choose depending on the virus, we have to choose which level we need to use. And it's also true for bacteria. Okay, it's also true for bacteria. Say, for example, if you want to work with bacteria which easily transmit through like air, you have to use a safety level 4. Okay. So, for example,
So there's different safety levels, right? Are safety level কোনটা?
Safety level 4. Yes. Okay, BSL4 is the maximum safety level lab. Okay. So, in each safety level, the measures to protect the us increase. Okay, and in this kind of safety level, we not only have to protect ourselves, also we have to protect the environment. So, for example, say you are culturing COVID-19. COVID-19 can spread through air. So you have to be protected when you work in the lab. Also, the lab have to be that secure that no air can escape from the lab to outside. Because if the air can escape from inside the lab to the outside, it means the COVID-19 can also escape the lab. Okay, they can spread in the environment. So safety level, ধরো যে সেফটি লেভেল 4 এ যে এরকম সেফটি থাকতে হয় যে ভেতরে যে কাজ করবে সেও কমপ্লিটলি সেফ থাকবে এবং ওই ল্যাব থেকেও বাইরে কোনো কিছু যাওয়া যাবে না, গেলে বিপদ, রাইট?
জি স্যার।
Okay, so you will learn about the safety levels in the near future. Okay, or you can just Google. Hmm?
আমি
Yeah,
কি কোভিড-19 কে সেফটি লেভেল 4 বলবো?
I mean COVID-19 case safety level 4 বলবো না. I mean the lab where we want to, if we, I mean, if we want to culture COVID-19, we need a safety level 4 lab.
জি স্যার।
Okay, COVID-19, we don't name COVID-19 as safety level 4 virus. Okay,
ল্যাবটা সেফটি
the lab, the name of the lab is safety level 4. Okay, it's not safety level, it's BSL, biosafety level 4. Okay, so you do one thing. Okay, so after the class, please Google biosafety level. So you will easily find out that. Try to get some idea there. I mean, as you are going to be a microbiologist, so it's good to know. You should, I mean, have idea about this information, that what is called safety level. Okay, so what is the difference between safety level 1, safety level 2, 3, 4? Okay, now I have a question for you. So you said, okay, the safety is actually here. Okay, I'll come. So I have a question for you. In Bangladesh, there is no biosafety level 4 lab. In fact, there is only 1 or 2 biosafety level 3 lab, I guess. Okay. There is no biosafety level 4 lab in Bangladesh, and also very few, maybe 1 or 2 running biosafety level 3 lab. But several diagnostic lab, they are detecting COVID-19, right? I mean,
জি
they're detecting COVID-19 means they're dealing with the virus. Then how they're doing it?
স্যার illegally? I don't know.
No, legally. They're doing it legally. But
স্যার ল্যাব নাই হয়তো বাট মানে এই যে মানে এগুলো কি বলবো আমি
হ্যাঁ,
কিছু মেজার মানে কি বলবো ইনস্ট্রুমেন্ট কি মানে যায় বুঝতেছি না কি বলবো এটা ওয়ার্ডটাকে মানে এগুলো
আচ্ছা, ইনস্ট্রুমেন্ট আছে, কাজ করার মতো ইনস্ট্রুমেন্ট আছে। আচ্ছা, আমি কোয়েশ্চেনটা একটু সহজ করি। হ্যাঁ, দে আর ডুইং ইট লিগালি। ওকে, there is nothing illegal out there, but there I mean, and there is no safety level 4 lab in Bangladesh, and but they are I mean, the labs who are detecting the COVID-19, that is legal. Okay, it's legal and it's okay to do that. There is nothing wrong with that.
ইনস্ট্রুমেন্ট গুলো নিয়ে কাজ করতেছে মানে আর কি নিজে সেফ থাকতে পারে ওইভাবে মানে থেকে কাজ করতেছে আই গেস মানে যেহেতু নাই ল্যাব তবু করতেছে কিছু তো প্রিকশন মেনটেন করতেছে তা কি বলতে পারতেছি না
Okay, আচ্ছা ঠিক আছে। আর কি, anyone else? Okay, I'll make the question more easy. Why we don't need safety level 4 lab to detect COVID-19? Okay, we need the safety
Sure.
level 2, but not 4. Yeah, please.
Is it because there's a written guideline about how to follow level 4, so that if you follow that, you can be legalized to do so.
Okay, I mean, there are guidelines. I mean, what kind of lab we need to use to detect COVID-19. So according to the guideline, BSL2 lab, safety level 2 is enough for detecting the COVID-19. We don't need safety level 4 to detect the COVID-19. Why? I already
Sir,
almost answered the question. Yeah.
স্যার যখন আমরা মানে when we detect this virus আমরা হচ্ছে একটা কি বলবো ওটাকে পাত্রের মধ্যে ওটাকে নিয়ে যাই যেটা কনফাইন্ড থাকে, সেটা নেওয়ার পর আমরা একটা কনফাইন্ড মেশিনে দিয়ে দিই। সো এটা আমাদের কালচার করা লাগতেছে না, এটা আমাদের খুলে কাজ করা লাগতেছে না তার জন্য।
Okay, almost. You guys are almost right. Thank you very much. Yeah, you really did very good guess. Okay. তোমরা আসলে খুব চমৎকার গেস করছো। Yeah, I'm really impressed. So well done. Well, the thing is, yeah, we are not culturing the COVID-19 here, right? In the
Yes, sir.
diagnostic lab, we are not culturing the COVID-19. So how they are doing it? They are collecting the sample from the patient. Okay, after collecting the sample, they are putting it into a tube, a closed tube. Okay, then they carry the tube into the lab, and in the BSL2 lab there are a special space. Okay, we call it safety cabinet. Okay, safety cabinet is it's a surrounded place, and inside the safety cabinet, it is like that. No air can come out from the safety cabinet. I mean, the safety cabinet is designed in a way that no air come out from the safety cabinet. মানে সেফটি ক্যাবিনেটের ভিতর থেকে কোনো এয়ার বাইরে বের হতে পারে না। Okay, so what the people do, they collect the sample from the patient and then they take this sample into that safety cabinet, and inside the safety cabinet they just kill the virus by adding some reagent. Okay, so they are not culturing the virus, so BSL-4 is not required here. If you want to culture virus, we need BSL-4. But as we are detecting just diagnosis, we are not culturing the virus. We are taking the sample from the patient. We are taking that. Then after collecting the sample, we are taking that sample inside the safety cabinet. In the safety cabinet, I mean the working person, they don't enter the safety cabinet. Only their hand. Safety cabinet ভেতরে শুধু তাদের হাতটা ঢুকা দেয়. Okay. So and then inside the safety cabinet, we just add a lysis buffer. It is called lysis buffer. Okay. We add a reagent which completely kill the virus. Then we use that killed virus to detect it. Okay. So that's why we don't need biosafety level 4 lab. If we just have a safety cabinet, that will be enough. Okay. Safety cabinet also have labels. Okay. Like be a safety cabinet level 1, 2, 3, 4. So for COVID-19, we need safety cabinet level 2. So that's why we don't need a BSL-4 lab to detect the COVID-19. And for detecting the HIV, we don't need any safety precautions at all. We don't need a special machines like safety cabinet. We don't need any safety cabinet for detecting HIV. I mean, if you want to detect HIV, just collect the blood from your suspected patient and then just detect in the open bench. You can do anywhere. Even we don't need biosafety cabinet, safety cabinet, because HIV cannot spread through air. Okay, বুঝছো তো নাকি, right?
Yes.
Okay. Well, basically, this last slide it talks about those safety concern regarding handling of viruses. Okay. So basically, there are guidelines. So like, what kind of safety precautions we should use when we deal with virus, and for what virus, what level of safety precautions we should deal with. But those guidelines are based on the characteristics of the virus, right? So, okay, in the guidelines, it is written that if we want to culture COVID-19, we need a safety level 4. If we want to detect COVID-19, we just need a safety level 2 lab. Okay. If we want to culture HIV, we need safety level 3. If we want to detect HIV, we don't need any safety level. Just the safety level 1 is enough. Safety level 1 means there is no safety at all. Okay. So these are written in the guideline. But as a microbiologist, okay, or as a infectious disease people, we have to know why, right? গাইডলাইনে সব লেখা থাকবে, কিন্তু আমাদের তো জানতে হবে কেন, ঠিক আছে? ওকে, আচ্ছা, আর 1 minute. Okay, just give me 1 more minute, then today's class will be finished. I just want to give you one idea about this slide. Okay, the basic of viral replication. Okay, this is about the replication or multiplication of animal virus. Okay, but before talking about this slide, please remember one thing: the basic steps of viral growth is same for all viruses. I mean, like the animal virus, the plant virus, bacterial virus. Basic steps are same. The virus infect the host cell, then inject its nucleic acid inside the host cell, and inside the host cell, the viral DNA can enter into the lysogenic cycle or lytic cycle. Okay, and if they follow the lytic cycle, they will synthesize their materials inside the host cell, and then after maturation they will get released. Okay, these basic steps are same for all the virus. There are just slight differences in animal virus or plant virus. So for example, for animal virus, what happened? So this is the virus, and these are real photo. Okay, please keep in mind, this is not cartoon photo. These are real photo taken by high power, very powerful microscope. Okay, so this is the cell, this one. Okay, this is the cell, this is outside the cell, this is the virus. First the virus get attached with the animal cell, okay, cell surface. Then the then the whole viral virus, they enter into the host cell through vesicle formation like this. Okay, I mean the host cell, they surround the virus and engulf it. And once the virus is inside the host cell, everything of the virus except the nucleic acid is gone, digested, destroyed. Okay, so only the nucleic acid it survive, and then this nucleic acid, they get multiplied by using the host machineries. Okay, so that's it. So what happened? Okay, another thing is, in case of bacteriophages, when they get released from the host cell get lysed. Host cellটা disrupt হয়ে যায়, এবং when the host cell get disrupted and the bacteriophage, they get released. But in case of many many animal viruses, what happens? I mean, when the virus are matured inside the cell, when they get released from the cell, they kind of take a part of the animal cell. I mean, okay, so think about the opposite of this one. Okay, here the virus, they are entering the cell like this way. Okay, is there any photo like the? Okay, here it is. Okay, so in many animal virus, when they get released from the host cell, they release this way. This is the cell, this is the plasma membrane of the host cell. Okay, look how they are getting released. When they are getting released, they get a part from the host cell. Okay, the extra layer. This is actually the envelope, right? So we know that many virus, they are envelope virus. Basically, they get the envelope by this way. Okay, this one is a cartoon photo. This is a real photo. Alpha virus, these are the virus, they're getting out from the host cell. They're getting out from the host cell. Okay, that's all about virus. Today's lecture is finished. Anyone have any question? কারো কোনো কোয়েশ্চেন থাকলে করে ফেলো, আজকে ক্লাস শেষ। এখন হচ্ছে মানে আমার লেকচার দেওয়া শেষ। I mean, I'm done with my lecture. If anyone have any question, you can ask.
স্যার আমার এই লেকচার নিয়ে কোশ্চেন নেই, বাট স্যার একটা কোশ্চেন আছে।
স্যার আমি হচ্ছে
হ্যাঁ, এই মামা তুমি আমাকে মেল করছিলা গতকাল?
জি স্যার, আমি কুইজের মার্কটা এখনো পাইনি স্যার।
ও আচ্ছা, তোমাকে একটু পর পাঠিয়ে দিচ্ছি। হ্যাঁ, ক্লাস শেষ করে। এই একটু পর means you will get within 2 hours. Okay, তোমার মেইলটা আমি দেখছি, কিন্তু তোমার খাতাটা চেক করার আমি টাইম পাইনি। 5 মিনিটের কাজ, আমি জাস্ট এরপর আমার একটা মিটিং আছে, তো আমি মিটিংটা শেষ করে তারপর পাঠিয়ে দেব। ঠিক আছে?
ওকে স্যার, ওকে স্যার।
স্যার আমাদের লেকচার কি চেক করা শেষ?
নোপ। না, আমি আসলে হয়েছে কি, থিসিস স্টুডেন্টদের নিয়ে এত বেশি ব্যস্ত ছিলাম এই কয়েকদিন, মানে তোমাদের অনেক সিনিয়ররা আটকায় যাচ্ছে আর কি। ওই প্যান্ডেমিকের কারণে ল্যাব বন্ধ ছিল, এদের হচ্ছে ওই থিসিস তো বন্ধ ছিল, কিন্তু এখন কোর্স শেষ, থিসিস না হলে তো ডিগ্রি নেওয়া যাবে না।
অনেক
তো
প্যারা।
লাস্ট 3 উইকসে আমার বেশ কয়েকজন থিসিস স্টুডেন্ট ডিফেন্স দিছে আর কি, এদের নিয়ে একটু সময় চলে গেছে, বাট থিসিস শেষ, সো নাও আই এম কাইন্ড অফ ফ্রি, সো উইদিন ফিউ ডেজ ইউ উইল গেট দ্য স্ক্রিপ্ট। ইয়া,
থিসিসের কাজ কিভাবে চলতেছে এখন এই যে প্যান্ডেমিকে, মানে অনলাইনে কিভাবে চলতেছে?
থিসিস চলতেছে না, বেসিক্যালি হচ্ছে ইয়া করতেছে আর কি তোমার, আই মিন দে আর রাইটিং রিভিউ।
স্যার এটা অনেক কষ্টের না জিনিসটা, যে মানে এতদিন ল্যাবে কাজ করার পরে যদি শুধু রিভিউ লেখে, ল্যাবে কাজ করতে না পারাটা একটু খারাপ লাগে।
Definitely, that's unfortunate, right? So, like, I mean, I always prefer that you guys do some lab work as your thesis. I mean, do some weight work. Weight work means working in the lab. I always prefer to that as a thesis you should do weight work, but they have no other way, right? I mean, they have finished all their courses, and they need to graduate.
স্যার আমি খেয়াল করতেছি আমার বেশি না কোর্স বাকি নাই, আমি বলি কি ব্যাপার, কি ইউনিভার্সিটি শেষ হয়ে গেল, পরে দেখি সিওডি কি শুধু সিওডি হলে ভার্সিটি ঘুমাইতে হবে।
সেটাই আর কি, এখন দেখো অসুবিধা নাই, মানে লেটস প্রে। ওকে, সো হোয়াট উই ক্যান ডু, উই ক্যান প্রে, রাইট? সো মানে তো যত দ্রুত সিচুয়েশন ভালো হয়ে যাবে, আমাদের ল্যাব গুলো ওপেন হয়ে যাবে, সো উই ক্যান স্টার্ট আওয়ার রিসার্চ अगेन, আদারওয়াইজ ইয়া।
ভার্সিটি যেতে চাই স্যার, আমার বাসায় ঘুরে এই বাসা থেকে বোর হয়ে গেছি।
সেটাই। আচ্ছা, এনিওয়ে, কে যেন একটা কি কোশ্চেন করতে চাচ্ছিলা?
জি স্যার।